we're going to move directly to the panel discussion. So I'm going to call to the stage Ron Bartek from the Friedrich Ataxia Research Alliance. Ron is co-founder and president of the Alliance. We've been working very closely with Ron and Jennifer Farmer and RDCA-DAP as part of a Friedrich Ataxia work group called FARA Ltd. Welcome back to Michelle Campbell. I'm going to welcome Collin Hovinga to the stage. Collin is a PharmD and a recognized expert in pediatric pharmacology. He has held appointments within FDA, peripheral and central nervous system advisory committees and several pediatric projects for the NIH. Welcome, Collin. Kevin Krudys, please join us, team leader for the Division of Pharmacometrics at the Office of Clinical Pharmacology, FDA. And finally, welcome back to Terina and Amanda. All right, everyone. Unfortunately, we couldn't get to Megan's presentation, but Megan has been doing a lot of groundwork, really leveraging the database we have for Friedrich ataxia, which is the biggest database that we have for that disease in the world, a database that is still growing. So Megan really worked on developing the models of disease progression, really taking a look at different endpoints and see how they track Friedrich ataxia. All these endpoints track Friedrich ataxia patients throughout their disease journey. So a lot of work also done by FARA on that. Megan has worked on a context of use for potentially further develop her work into a clinical trial simulation tool, such as the one that you've seen in Terina's presentation. Megan also was leveraging the database that we have. So I mentioned PSP, progressive supranuclear palsy. It's a rare neurological disorder. So she really started, we have really good trial data for that disease. So she really started working on that disease as well, and similarly developed some models of disease progression. So she was just going to show you some early insights into what we can do already internally within RDCA-DAP to leverage the database. So that's about what you were supposed to see.
Well done.
Just to start, I think I'm just going to start with you, Ron, since you've been, following Friedrich ataxia effort and following the C-Path effort very closely. How did RDCA-DAP help you as a foundation collecting data, really leverage, this data and use that to inform drug development for decision for the disease? And overall, what impact this work had for the foundation in the process?
Thank you, Alex, and thank you for that kind introduction too, and filling in the gaps in Megan's fine presentation. I'd like to begin though by saying how thrilled, honored, and privileged I feel, and how oh no, not again, and how FARA feels the same for just being included in this panel and to be so deeply involved and engaged in the RDCA-DAP and the critical path and NORD and FDA effort in our behalf. And I also want to recognize and thank my dear friend and colleague, Jen Farmer, whom Alex was kind enough to mention, the FARA CEO, and for her helping build, nurture, and grow the natural history study from the very beginning, back almost 20 years ago when we started this this effort that's proven to be so instrumental in our progress. Now I'll answer Alex's fine question. So, the question really is, how does RDCA-DAP help? And as Megan was already saying and was about to say more about, the RDCA-DAP and the C-Path efforts that Megan outlined are already helping. We're making real, genuine progress, and we look forward to much more and the potential for the clinical trial design model and the further development of the disease progression model. But we're already making tremendous progress in both regards. First, because the program has helped us facilitate integration and standardization, no mean feat for these huge databases. And so integration of all those data and standardization with common data elements and so forth, and integrating into the natural history that we'd spent more than 15 years in developing, along with the placebo-arm data, more natural history, if you will, from 6 clinical trials, 6 different industry pharma partners, and not only helping us integrate and standardize those data, but helping us provide access to those data. We've already had more than 20 requests for access to the database. You know, we've achieved sort of a critical mass of those data that we're already being able to collaborate with our own analysts and C-Path analysts, even FDA analysts in in assessing those data towards endpoints, towards analysis of clinical trial protocol design, clinical trial results, even analyzing clinical trial results and comparing clinical trial subjects to very carefully matched natural history subjects, so as to provide potentially external controls for the trials themselves and external controls for analyzing additional data that could be presented as confirmatory evidence in support of positive clinical trial results. So we're making tremendous strides already, and we're looking forward to even more. So I'll stop there.
Thank you, Rob. Yeah. Jumping on some of the things you said about standards, and wanted to ask you, Kevin, that question. You know, RDCA-DAP really tries to provide a structure with standardized data, harmonization of data. Why is it important? And maybe from your own perspective at the agency, why is it important to have a place for that?
Sure. I think the importance is for us to have the confidence in the quality of the data. So if you think of the standard approach that we typically see, is that perhaps a sponsor might have a publication where they have a reference of some source data, and they include that, say, in a meeting package or in a submission. Then we have to have a look at that and say, you know, why did the sponsor choose that? Do we have the access to the source data? If we don't, the sponsor might have to get that and send it to us, and the format might not be something that we can work with. So if you have a platform where we have the confidence in the data, and a sponsor could actually say, you know, we went to the platform, we filtered it by certain factors, and here's the data, here's the code. That's something straightforward and simple for us to have the confidence to go forward and say, okay, we can look at that, and it can speed up the process, as opposed to taking, you know, a long time to get the data or to talk back and forth with the sponsor. So it's really about the speed and the confidence that we can have in the outputs of these things. And I think you made a comment about, you know, it's more work for us. You're talking about the tools, but it's probably not more work for us. It's a lot more work for us if we have, you know, 4 sponsors that are each coming and each have their own tool and endpoint and their own source data that they put together to inform the tool. That's a lot of work for us. If we have a standard approach, which is transparent, it's been worked on across sponsors, it's pretty competitive. That is something that actually could help us in our job as we go forward.
Yeah, no, thank you. Great to hear that we can facilitate a lot of the work that you're doing. I'm just gonna go back to something more focused on patients. You know, maybe you all can answer that, like really what is the impact of these drug development tools that we're working on at C-Path to patients? So maybe I'll start with you, Michelle, if you want to give a general comment, and maybe Terina, if you can like make some precision to, you know, just talk about the specifics of these trial simulation tools that you've been working on a lot within your consortium.
Yeah, thank you, Alex. And so I am gonna stare into the giant sun because if Ron and I look at each other, we've done panels before, we typically make each other cry in a good way, because we're always celebrating the successes of why we collaborate together and why working in the pre-competitive space is so important in the advancement of patient-focused drug development. And so I was thinking about this during Ron's talk and what Kevin just said, and I really want to thank the C-Path colleagues for presenting those examples, because people want to know, what can my data do? And here's a potential 4 examples of what can be done when we have sufficient amount of data. I think that's really critical. We have to have a sufficient amount of data, and that will take some time to develop that. But that shouldn't mean you should not consider contributing data, because we need to build that volume to be able to pull. And I think about how this is impact PFDD. Well, in the end, this is part of patient-focused drug development, right? We are being able to perhaps include a broader population into a trial, because we've got past data that is suggestive that perhaps we may need a broader population. You know, and only that will happen through modeling and the clinical trial simulation tool that was described. I think it goes to Ron and his effort to take on and build a natural history database and evolve it over time, and be willing to make those changes to bring in and spearhead that. So I think the patient is always the forefront, because number one, it's their data. But they're always in the forefront in terms of the end goal is to always, the patient is in mind. How can we better our patients who are going to participate in our clinical trials through the opportunities with data sharing, and be able to use these advanced analytics to help us better inform. So while it may not be as clear to many why data sharing is important, how it does impact patients, it really, going back to the question I was asking me earlier, you know, about patients being on the forefront. Patients are always on our forefront, and this is just another way of bringing that patient voice in a way, because it's their data that they contributed. Just another way of being able to analyze the patient voice through pulling this data together and being able to use advanced analytics to help inform rare disease drug development.
Could I just reinforce everything that my dear friend Michelle said? It's so important. I mean, there's so much emphasis in patient-focused drug development recently on, you know, doesn't that start when the FDA is reviewing my application and we can raise the roof and make the voices heard? That's not true. You've heard several FDA speakers like Teresa and Billy talk about the importance of early engagement. This is the way the patient gets engaged early, is doing the natural history studies, going into the clinic once a year and for 15 to 20 years, and making their voice, their data heard. So, you know, sorry. So this is patient engagement early and often. And, you know, through this program, we're now able to put all that hard work to good effect. And so bravo, you know, the patients that participate.
Thank you, Ron. Terina, do you want to comment
Sure.
a bit on
Yep.
specificity of our CTS, clinical trial simulation tool? Sorry.
Happy to expatiate on that. It's really, you know, echoing a lot of the themes that we've talked about already, and Michelle has highlighted. I do agree. Sometimes when I'm asked, or when I, for example, earlier this year when I was at one of the meetings for the Duchenne muscular dystrophy community hosted by PPMD, the patient advocacy group, there were patients and families everywhere. And when they met me and we had the opportunity to interact, I would explain that I'm at the Critical Path Institute and I lead this consortium. And then they would ask what I was doing. And I'm explaining to an 11-year-old the lost in translation aspect of explaining to an 11-year-old what we're doing with their data and simulating this tool. I was like, you know what, let's just make it like video games. You know, this is a way to, you know, to reach that particular audience. But what I think is really key is making sure that we really pull back a little bit and bring some objectivity to these projects that are extremely detailed, highly esoteric, to really say how many ways can I articulate this and connect it directly to either the individuals who participated in the study and contributed the data, to the data sponsors or the owners of the data who, you know, provided it to us. That's really what I try to focus on, is being able to take this clinical trial simulation tool that's been years in the making, and find those multiple connections to the community that provided all of the enabling steps to get it where it is.
Thank you, Terina. I heard, you know, Michelle talk about, you know, the amount of data that we need. And, you know, when we saw Amanda's efforts with, you know, cohorts of thousands of patients and you know, Duchenne as a huge database, just wondering if, Collin, you would want to comment a bit about, you know, what can be done in other rare disease where there is more scarcity of data. Are there specific tools that we can consider to still like make some progress while more data is arriving? And maybe Kevin, if you want to comment a bit to that aspect.
I think some of it is really looking at trying to.
I think just sharing you up. Sorry.
Sorry. Yeah. Go team. I think that, you know, one of the things that is really important to do is interface with the community and the investigators to find sources of data and information. You know, in most people are once it's been demystified, people generally are more comfortable with sharing their information. Electronic health record strategies are always fruitful, but outreach to the community is going to be essential for this to be successful. One of the messages I haven't heard here, because I'm a trial kind of planner person, is a lot of these rare diseases don't have a lot of patients. So if you're informing in the community that you're trying to make best informed decisions about how to design a clinical trial that will begin to get, you know, stakeholders involved. I don't know a family that said, I want to sign up for the placebo arm in a clinical trial. So these tools are really essential to be able to maximize that or stay in a trial longer than they have to for a therapy that might be beneficial. So anything that can, you know, step into, you know, communicating that to the community also will engage people and, you know, helping them become involved in submitting their data.
Yeah, so I think if you share data, you start to learn what you don't know as well. So I think we've had that experience in a few consortia. DMD is one example where we were pulling the data and looking at progression, placebo response, and patient dropout. And from that, I think we learned that we don't know much about the placebo response in the trials or patient dropout. And that could help you to plan in the future what kind of trials or data sources are important to incorporate, or if you're starting a new trial or study, what pieces are important to add or to measure from patients to add, so that as that data is being added, you can learn the important things that were not apparent as you started the process.
Thank you. And I guess I'll add one answer to my question. There's also, you know, the diversity of rare disease with the diversity of needs and information that is lacking, and always insist on that. Cross-sectional data can be very valuable to some communities that are barely starting their collections effort, even barely starting to emerge as a growth foundation. And I think being able to display simple genotype or phenotype or cross-linkage genotype to phenotype on the platform is also very important to some of these communities. Amanda, at the end of your presentation, and you told that to us also internally, you seem to want to start to engage in growing a bigger database, which, you know, you had access to data to do the work you conducted, but now you want to take one step further. So just wondering if you could comment a bit on the why. You know, you see that as very relevant to take your work to the next step.
Testing. Great. Yeah, so when we started the process of collecting data to support the iBox regulatory submission, it was focused on what are the variables of interest to calculate an iBox score, but then also thinking about the practical application in a clinical trial and what you would need to show to provide the confidence to regulators for the different diversity in the population. So with that learning, and I'm going back to the iBox and then I'll talk about the future. One of the ways that we reassess the iBox is, like I discussed, the adapting it for a clinical trial endpoint, but also thinking about, do we even need biopsy in the model? Biopsy is an invasive procedure. It comes with risks, so it's not fun for patients. And then if you think about a sponsor and the design clinical trial, the additional effort and complications and dropout that go along with patients not getting a biopsy. So one of the things we did was explored how the model would perform without biopsy input. And we saw that it performed well. And so this is one example of how we incorporated feedback from the community, both from patient perspective, providers, but also industry, to align to support. And at the end of the day, the idea of clinical meaningfulness goes back to what does the data show, and is there any literature precedent to support that? And the data, you know, supported it for this. So now that we have a extensive database of over 25,000 kidney transplant recipients across the globe, our vision is exploring how can we create a tailored data collection form based on variables of interest that can support future regulatory needs that aren't just based on one, you know, regulatory effort like the iBox, but creating a list of variables that are important to the transplant community, both patients, academia, and regulatory, and having this list come from retrospective data, but then also how can this be like a living repository and analytics platform for prospective? So this is our vision of, our first focus is kidney transplants, but being thoughtful about the framework to have a tailored data collection form to support future regulatory across the span of kidney transplant. And the other part is having engagement with the international transplant community. So transplant's unique in that every single patient who receives a transplant is accounted for in the United States. It's different than other diagnoses where, say, diabetes, you may have someone have diabetes, but they don't know they have diabetes. Whereas with transplant, we know all transplant recipients. So we have a unique opportunity to coordinate in an international effort to make this a unified data repository and analytics platform. When you think about other countries, everyone uses their own registry to help facilitate the improving patient practices in their country, right? And so how can we have one platform consisting of diverse data from across the globe based on a tailored data collection form? And that's living repository. So the idea to have it a central place to streamline review for any regulatory agencies, but specifically we think about EMA and FDA. This can be a tool that can help leverage and ultimately accelerate medical product development and get patients newer therapies. And for those who don't know, transplant, the easiest part of a transplant is a surgery. The patients have to take immunosuppressants the rest of their life. If they miss a dose, there's no other diagnosis that the risk is so high. If they miss a dose, they could lose their gift. And these side effects that come with the medications can be very burdensome. And you may not see it from the naked eye, but patients experience it. So the standard of care has been around since the 70s. The last drug approved in the US was in 2011, and it's not even the standard of care. So patients are desperate, clinicians are desperate for new therapies, and these regulatory endorsement, the one that's upcoming next month with EMA, we're going to get that qualification, is the first step to reinvigorate drug development and transplant.
I don't need the mic, but
No, I don't.
thank you. No, that was a great answer. Maybe Collin, do you want to reinforce a bit on the value of this work to accelerate drug development?
I think, one of the things that you just mentioned was, the complexity of the regimens that patients take. I mean, so many of the rare diseases and the conditions that have been described today, either you're ad hocing trying to figure out what to give a patient because either you need to add something that you don't to manage their symptom, or you're just suffer through a situation where, you have to go through and give medicines that are old. And so having more information, or at least the hope of new therapies in development, I think it shows great promise. I get excited when I hear these things because it's really the opportunity to make impactful change. I can, as you were talking about there, I can remember the past, since I used to do kinetic service, having kids that failed transplants because their immunosuppressants, they weren't being monitored effectively. Their transplant drugs drew too low. They lost a heart, they lost a kidney. I mean, that's not a conversation that feels comfortable because the kid didn't like taking the medication because of the way, didn't want it, didn't for some reason, either a side effect or because they didn't feel comfortable with it or didn't feel they needed it anymore, because kids use oftentimes adherence is, adolescents in particular. So having new therapies that, might be more attractive and promise hope, I think are really, these tools are the first step in doing that.
Oh, thank you. Without being too technical, I can't, but, Sergey works for SCD, and so there are several gene therapies in the pipeline. So I'm just wondering if maybe Michelle or Terina, if you have some insights into that. What are the particular needs in terms of the kind of work that, when we talk about the kind of work we do, working on endpoints, working on disease progression modeling, what are the particular needs for drug development as it relates to gene therapy specifically?
Stab, I am not CBER or gene therapy expert, but I would say that any type of data collection that can help inform any clinical trial, gene therapy, a drug development trial is going to be informative. Those trial designs look different. And so I think any type of work, such as work that the Duchenne consortium is doing and some of their trial simulation work, and I know this has come up in the consortium, is to start looking that way, will always be informative. And I think it will be, hopefully a great addition. Those are new and novel trials, so we don't really have a lot of data probably to help inform some of that. But I think that we can use lessons learned to help inform the conversations that would happen with my biologics colleagues. But I do think that the nice part with data sharing in the platform and these analytic methods, can expand across different types of trial designs and different types of products as well. So not just ones that CDER regulate, but perhaps our CBER colleagues and what they work on. It is really maybe treatment agnostic per se. I mean, while our focus is on drugs and what CDER does, we do recognize that there are diseases and disorders that do have the genetic component, and so that work may occur at some point for gene therapy. And so I think these analytic opportunities will still be beneficial for that as well.
Yeah, and just to piggyback off what Michelle said, I think when there's a tendency anytime, the rare disease space, of course, is susceptible to this. When you identify a monogenetic cause to disease, there's a tendency to think, okay, gene therapy will cure it. The number of steps and requirement to get from identifying the gene to a cure with gene therapy are just massive and complex. Some of, certainly we could have a panel all day on these topics. I won't do that, but just to highlight a few of the key challenges. As Michelle said, designing a gene therapy trial is completely different. There is no placebo because you can't have, you can have, you can't do like a sham gene or do a different gene, for example. You don't have washout because it's a permanent modification. So you have to be very careful with your safety because you can't stop dosing once you've dosed. And then even just some of the other considerations within the field of Duchenne muscular dystrophy, the challenges that the companies that are pursuing gene therapy approaches are facing are immune responses to the viral vector that's used to deliver the gene payload. And this is because it's an adeno-associated virus. So some children who have colds could have an already high antibody inherent response that would make them react poorly to the gene therapy. So they have to be screened ahead of time. So you can imagine the impact on patients and their families when they are optimistic about enrolling in a clinical trial. They're ready to make the commitment, and they find out that because they have antibodies against the viral vector, they cannot go into the trial. The other challenge is because of that antibody response, you can't do multiple dosing. So find a dose that works for the entire musculoskeletal system in a one-off. That's really hard to do also. So you can't do multi-dosing. And these are just a few of the complexities. What is really gratifying in the Duchenne field is that the companies that are working on gene therapy and are facing some of the same challenges are talking to each other, and they are working together to try to understand how to overcome these obstacles, and then anchoring it back to why we're all here. And what Michelle was saying, the data is what's really going to help them and us and regulators and individuals who are thinking about a gene therapy trial to move forward with better efficiency, because we need the data to understand how to improve, cloaked viral vectors that don't cause the immune response. We need data to understand, how to bring potentially an individual who's had a gene therapy but wants to try another therapy that's a small molecule. How do you incorporate them into a new trial design? So the data is really what is the common denominator for all of these considerations and many more that we didn't even really address.
Just love to reinforce everything you just said, Terina. Because gene therapy is unique, as you mentioned, it's once the gene is introduced, it can't be removed. It's not like a small molecule, we'll stop dosing. And so that drives several important things. So you mentioned the importance of, trying to get away from strict placebo control, which introduces the terrible importance of natural history. As an external control that might be allowed to replace placebo control. I know there are real concerns about that, but it might be one of those instances when if you can't do a sham effectively, that maybe external controls would be important. Another important element is that there will be additional temporal requirements, because you introduce the gene, you don't know for how long it'll be maximally expressed, or does it taper off? Is there a plateau? And along with the protein, and do the toxicities fluctuate over time? So there'll be a lot of preclinical and clinical requirements by the agency that are understandable. So, but again, the natural history involved in this platform can really be instructive on all those fronts.
Amanda?
I find gene therapy very fascinating, and I've been reading the FDA guidance documents. There's been a lot of FDA guidance documents that have come out the last couple months, especially trying to help sponsors with their clinical trial designs. But going back to the idea, some of the big considerations that I've been reading about is about the natural history and not having adequate knowledge of natural history. So this goes back to, let's start getting that data now that we can have it yesterday, it's yesterday, today's today, but as early as you can. And then the other part is the analytical validity, whereas, gene therapy and the manufacturing processes for it are complex and aren't as well defined, and harmonized. And so it's a newer area than we're used to, say, for renal function. We know serum creatinine lab, we can trust the assays and whatnot. So it's a different level that sponsors and review regulatory are having to work through. But those are the two big things that I've read about.
Thank you. Before I open the questions to the audience, I'm just gonna end something that we talked with Ron, but I think it's a question for everybody again. The platform we're developing this unique format, and we're trying to develop, potential for cross-disease discovery using the tools of the platform. But at a more high level, in terms of, all this knowledge that we develop, these tools, what is the potential that you see from RDCA-DAP having to be a helpful tool and program to have lessons learned and apply lessons from one disease to the other? So I know Ron wants to talk. Collin, also maybe coming from your experience in pediatrics, where there's a lot of ongoing work that relates to extrapolation of adults' data to pediatrics. Like, how do you see maybe RDCA-DAP being able to help inform pediatric disease overall? Ron, you can start.
Okay. I'll start by saying FARA is thrilled to hear from, I think it was a portion of Megan's presentation you didn't get to hear, was how lessons learned from the FA database are being applied already to PSP. And we're thrilled to hear that. We're also looking forward to the reverse, in which we can benefit from insights from others. I mean, there are so many things that we all need to know. There's so many things, especially neurological diseases. So many of our rare diseases are neurological. We don't know how to measure neurological impact and benefit in affected tissues. I mean, so many of our biomarkers are based on blood. And blood's not an affected tissue in our disease. We need to know how to measure impact on neurons and glial cells. And, so if anybody comes up with solutions about how to measure impact in neurological tissues, spread the word and put it on the platform. There are all kinds of other endpoints. I mean, we've got so much in common. The common neurological examination is an integral part of what you heard about from Megan, the modified Friedreich's ataxia rating scale. So many of our diseases start with the clinician doing the neurological exam. If we can refine endpoints based on those neurological exams, we'll all benefit. So again, if you've got solutions to those, get the word to the RDCA-DAP and let's go.
First comment that comes to mind is just the across diseases, particularly in pediatrics, is the standardization, because the language that we speak, even in different jurisdictions, units of measurement, how things are done, are pretty diverse. So coming up with a common data dictionary that can be reflective across multiple diseases in different age groups, is a very important step in and of itself. I think the second part of it is that, one of the things that I used to get was a lot of comments from sponsors about what I would say are common things that are issues. And they're like, normative things about, how do you assess maturation, or how do you look at neurocognitive change over time in different disease states? So coming up with sort of standard ways to approach these common problems, because they affect so many drug classes and so many disease states, and there's really not a predefined strategy for how to iron these out. So when you have this, it's in a bit, I guess, disease agnostic tool in a sense that you're looking at from a high level, it provides an opportunity to start thinking strategically about, how would we look at these things, what similarities and what differences there are in diseases, and sort of come up with, more, I guess, more sensitive ways to look at, common developmental pathways with respect to drug therapies.
Great, Ron. Michelle also.
I was just going to add, so I really appreciate Amanda's enthusiasm about the future vision of what the transplant consortium can do with the standardization of iBox. I think that's the enthusiasm we want, because that is a lesson learned, right? How standardization, hearing that we've got ontologies being worked on. These are things that are really quick, that are really critical, because then if we can learn them, standardize them, and we can get them back out into the community, into our stakeholders, and have them starting to be uptake into clinical trial design, CRF forms, natural history study designs that groups are doing and collecting data, it's going to benefit us better in the long run when it comes to data interpretation, migration, all those things we want to do. So I think that's important. So I think we need to be very open to lessons learned, or adapting over time if there's been a change in the standard of care. And we need to start capturing that, in our databases and natural history studies to make sure we're reflecting current status. That's really informative to us when we want to make sure if we want to use as a potential comparator, we need to have the most accurate, up to date as much as possible. Obviously, there are always constraints in some of these cases, but I think lessons learned are always a great thing. I think we have definitely learned some through this initiative. And I think those who have been pioneering data sharing in general have definitely got some lessons learned. I think we had a conversation broadly brought up the part about informed consent. So if you've ever had to do informed consent process, I've done it plenty of times, it's a process, but every patient I ever talked to, or every caregiver, I should say, because my patients were typically not able to, or pediatrics, so they were not providing consent. Some of them were able to provide assent. Every caregiver said, I'm willing to help the next patient. This may not help my child, because some of these were not true drug interventions trials, but I can help the next patient. And so I think we shouldn't be afraid to have these conversations and making sure we're building in these conversations, because I know the families want to feel like they're contributing to the science. And I think that is part of that educating about whoever's idea about starting small and early and introducing to families, because I've always had a positive encounter with patients and families when it came to recruiting in my lifetime ago, is that they do want to feel like they are contributing to the science. And that contribution will help us, will help RDCA-DAP, it will help drug development, may help evolve into something actually to be in a pipeline, right? So that's something we really haven't discussed, and that's an entirely different conversation for a different day about, those rare diseases that are fortunate to have active pipelines, those that are primed and ready to go. And there's a whole bunch, I know. And then those that may be a little bit farther behind, just because we need more of that information. And I think this is why this initiative is so great, because I think it can help those that may be further behind, and not in a bad way that they're behind. It's just we don't have that information to be able to have those conversations. And this type of platform and initiative can help us, I think, not only those that may have active platforms, but those who do not at this time, and maybe help spark some conversation to where to start approaching what a pipeline will look like.
Thank you so much, Michelle. This is really an exciting conversation to me. I think so much can be gained by all of us from this conversation. Two specific points that come to mind for me in this regard are the things we can share about two things. One is cardiac endpoints. I mean, so many people in this room and online suffer some form of cardiac issues in our diseases. In our disease, that's the biggest killer. About 80% of our patients die with congestive heart failure. Yet we don't have much in the way of endpoints on cardiac tissue other than mortality. And we can't wait to see how many of our patients live a year longer. And we need solid endpoints, at least biomarkers of cardiac benefit. Another area in which I think we really need endpoints that we can all help with is in form of trying to assess benefit in pre-symptomatic patients. If we always wait until the patient is clinically manifest, we're going to lose so many opportunities for therapeutic development. If we can develop the endpoints for monitoring failure and success, benefit and lack thereof in pre-symptomatic patients. Think of when the treatment window is broadest, think of the progress we can make for our patients. So if you've got ways to measure impact in pre-symptomatic patients, spread the word.
Thanks, Ron. Let's take a few audience questions.
Do I have to say my name again, or is it okay to keep going? Comment to questions. Ron, your passion is palpable, which makes coming here in person worth it. So thank you for that. Next question is Amanda, but anybody can respond. I completely agree, and I'm glad you both brought this up. Lessons learned are critical. There's no point in reliving prior mistakes. But Amanda, what could you have done for your project to have saved some time? Like, there's the lessons that you learned, but there are things you would have done differently that would have saved you some time. Can you comment on that?
Thanks for the question. So much of the process is really, the TTC was formed in 2017. So the first year is bringing stakeholders together, thinking about what the unmet need is, and creating a collaboration, a healthy collaboration. And the majority of time is spent not so much deciding what to pursue, but more so doing a landscape analysis of the data to support the regulatory qualification. And then it's getting the data. And the longest effort is getting the data, right? Like Alex talks about. So it takes time to build rapport with the data contributor. It takes time going through data sharing agreements and going through lawyers and all the details that go into that. And then it takes time to curate the data. And the easiest part is running the analyses and doing the writing. But for regulatory submission, especially for a surrogate and a composite, we were as efficient as we could be. So I think we did it. I don't think there's really anything else. I don't think there's anything we would do differently. So this is kind of part of the nature, I think. So yeah, I'm really proud of our team, and I know the data inside and out, and I work closely with the data managers when they curate the data. I go through it from the clinical perspective, and I called the manufacturers to get the assay information. And we have a really solid team. So I'm really proud of the work.
Okay. So
I'm sorry. I just want to give. I'm sorry, we're running out of time, so I just want to give Penny the opportunity to ask a question.
And a great question it was. Getting the data is such a key thing. And I feel like some of our spaces are in a bit of a catch-22, where until you have a ton of trial failures, there's also less than incentive to say, hey, this is going to be needed to solve some of these pre-competitive tools. But then you have to go back and get the data that was probably not consented originally for that purpose, unless you're really lucky or unless somebody was in the right place, which I would hope, and to me, this is a core piece of on the advocacy side, is that every trial should be getting designed with template informed consent that will enable that. But with the really amazing examples that you all shared just now, I'm assuming that some of these trials had been maybe not originally consented, or some of these natural history studies were not originally consented for this type of broad sharing. And maybe there was a reconsenting process, or maybe there's something about the RDCA-DAP platform that allows it. I'm just wondering if you could speak to the abilities of getting retroactive data for when something was not initially designed for that purpose.
I can give one data point, and that is that as we were working with RDCA-DAP to standardize and integrate our natural history data, we were also working against the new European standards for, confidentiality. And we had to reconsent our whole natural history study at the same time we were developing the data to put on the platform. So it can be done. We were able to do it. And we continue to conform to those new requirements.
Harder to do it when the people may have passed away already, though.
Oh,
right?
You couldn't do that, but yeah.
Yes. Yes, and then.
Oh, yeah, I just wanted to just
something.
do a quick follow-up to Penny's question really briefly. So you're right there, and actually within the Duchenne database that we have, most of that data is actually not available for general users to apply and access. It's a subset of our database that is available for that. And that's because of the shareability restrictions, which sometimes do predicate on the consent aspect. But this is an example of why getting the data into platforms where even if the wide population can't access the data, the scientists at C-Path can, and they can build these tools like we just did. So most of the data in that clinical trial simulation tool is not accessible to a wide data user, but it's still extremely valuable. And this tool will now be accessible to everybody. So there's iteration to that process.
Thanks, Terina. We'll take a last question, and then Billy, if you want to. Okay.
Thanks. Hi, I'm Julianne Mills. I'm from the Chan-Zuckerberg Initiative Foundation. And Terina, I just would like to ask you to maybe tip it off, but I'd like everybody's feedback from an organization's perspective that works to try to empower patient-led research. What are some of the things that you wish the patient groups had come to you knowing, or what kind of infrastructure do you wish that they had come prepared to share with you as part of that partnership?
Yeah, that's an excellent question. I think within the Duchenne community, we're very lucky that it's such a motivated group, and the patient advocacy representation is tremendous. I think that the parts that are really challenging, and this is what I think to the crux of what you're asking, what would be extremely helpful to us is if those of you who really are, right there lockstep with the individuals with the disease and having these conversations with them frequently, to help us, to help them understand, to do that kind of education and priming component of what it takes to be in a study, what it takes to be the difference in a study and in a clinical trial, to be able to help them understand that, the clinicians or the regulators or the sponsors who have exclusion criteria for a clinical trial are not purposely trying to exclude people. It's necessary to plan the trial in such a way that you can make interpretations. And so it's much easier if the patient community, through advocacy groups and other education formats, can be primed in those complex conversations before we get to the point of, having the other limitations that we do have for the data and the studies. Did that adequately answer?
Great. Well, thanks with that wonderful last answer. We're going to end up this panel discussion. I want to thank you. I'm sorry. Yeah, I want to thank you. Oh, you want to say something, Teresa? Oh, okay. So I want to thank everybody for joining. I had wrap-up comments, but Michelle and Ron, you did a terrific job at ending before the Q&A for me. I wouldn't have done a better job. So thanks, everyone.